Hello everybody, this is Dr. Lampert, and I am going to be recording the lecture here on protists. And as part of the lecture on protists, I'll be talking about where eukaryotes came from and covering the topic of symbioses. So you'll see I did the photos here again to all represent protists or symbioses, or for the most part symbioses of protists with something else. So let's kind of go over each of those. So I'll start with the bottom right. So the bottom right shows a fictional symbiosis, but I'm showing this to kind of give you an example of the definition of a symbiont. So an organism that lives with another organism. Staying in the bottom, I have a photo of a cat and a rat, Itchy and Scratchy. And Itchy and Scratchy kind of represent a long and, in this case, extraordinarily violent relationship between two mammals. And it turns out that this relationship between two mammals can also be influenced by a protist, specifically a protist called Toxoplasma. And Toxoplasma will be covered, if not in the lecture that I'm recording, at some point in class as we finish the protist concept. Okay, kind of continuing the clockwise look at the photos, we have a red tide. Red tide is a explosive growth, a bloom of dinoflagellates, which are one type of protist, and when they have when a red tide occurs, that can have pretty bad ecological effects, including bad effects on human health. And so we'll talk about protists and humans quite a bit here in the lecture. And speaking of protists and human health, the top right image, that is the #1 infectious disease of humans worldwide. This mosquito is potentially vectoring malaria, which is a infectious disease caused by a protist called Plasmodium. And there's actually 4 species of Plasmodium that can attack humans. The interesting thing about malaria, while we're looking at this picture of the mosquito, there's only one genus of mosquito that causes human malaria. It's the genus Anopheles. And so even though we have all sorts of horrible mosquitoes biting us here in the U.S., luckily we don't have Anopheles vectoring malaria to us right now. And this is a pretty remarkable photo too, showing a nice engorged female. So her gut's so chock filled with blood that it's actually stretching out and stretching her exoskeleton apart to the point where you can see her clear gut through her exoskeleton. So pretty amazing photograph there. Alright, so the learning goals, we do have a goal about symbiosis. We do have a goal where we should be able to all know where eukaryotes came from and understand how important symbioses were in the evolution of eukaryotes. There's going to be a goal to understand what protists mean, so kind of maybe a forgotten group compared to some of the others, but extraordinarily important. You'll notice that none of my learning goals are associated with taxonomy or classification of protists, identification of protists in any way. There will be some questions about protist groups in the lecture quiz, but we're going to mostly learn about the protist groups by an activity. We're going to have little presentations that I call the protist tournament in class to learn about protists. But I'll describe most of the information about protists here in the lecture too. And of course in lab we'll get plenty of good chances to look at protists and I'd be able to identify them in lab. For the reading we have, I'm kind of going to stop putting different sections since the two editions are so similar. So I'll just say is what we have in our Campbell biology textbooks. So chapter 25.3, or concept 25.3 of Campbell Biology has some important information about the evolution of eukaryotes, and in particular how symbiosis played a role in the evolution of eukaryotes from their prokaryote ancestors. And even though it's the same concept, the page numbers are slightly different. You don't even have to read the whole concept, just a couple sections about the evolution of eukaryotes. And the evolution of multicellularity is an important thing to read too. Chapter 28 is the Protist chapter. It's mostly a Protist diversity chapter. It has 6 sections, 6 concepts. And 4 of those concepts are going to be descriptions of Protist diversity. So you'll definitely want to read the whole section. There's 2 papers that I have posted for you to read. We may possibly get to a discussion of these in class. I'd like to. If we do, that'd be great. But these are papers regarding what I think is one of the most fascinating protists out there, and that is toxoplasma. Amazing little creature that causes pretty amazing effects on behavior of its mammalian hosts. A little bit more about what to read. One of the difficulties of learning protists is that they change so darn much. And using different textbooks, using different editions of the same textbook, they can be wildly different. You go online and look up protist diversity and look up protist groups. You might see extremely different things than you do in the textbook. I will say, as far as the protist classification of record, the one you'll need to know for a test and things like that, we're going to use the protist classification from the Campbell Biology text. So make sure you're aware, and my PowerPoint will have the classification from the text. Make sure you're aware of the classification in the book. Maybe some of you chose not to buy a book. Maybe you're just using Mastering Biology. Maybe you like to just look things up rather than look through a big old dusty book. But I will warn you, the classification of record, which might be very different from Wikipedia or other websites, is going to be the textbook. Okay, warnings aside, what's different between the 9th and 10th edition of the textbook for those of you that might have different definitions? In 25.3, concept 25.3, the 2 sections that you need to know are on different pages. That's not a big deal. I put the different pages in the on the previous slide. A couple taxonomic differences. Thank goodness there weren't that many taxonomic differences, but we did have some. So what's in the 9th edition, 2 supergroups. The chromalveolates and the rhizarians are actually combined in the 10th edition into something called the SAR clade, which is just the straminopiles, alveolates, and rhizarians. So it's kind of a cop-out. It's not much of a difference. The trees themselves look about the same, except you just have different things combined as a group. And what they recognized was that if you make a group of the straminopiles, alveolates, and rhizarians, there's actually evidence since the 9th edition came out, apparently. The 10th edition says there is evidence that those 3 groups can be combined into a monophyletic clade. In the 9th edition, it doesn't really look that way, but according to the 10th edition, there's some new molecular evidence that supports that. So that's a difference to note. And another note that I was really sad about was that the oomycetes, which is a very important group, kind of vanished off the face of the earth in the 10th edition. They are a group of straminopile, so they would be in the SAR clade if they were in the 10th edition, but for some reason they completely vanished. But at least in 2014 I missed them too much, and I like them too much that I can't really let us get away with not covering them. So we will cover oomycetes at least a little bit for the lecture quiz, and they'll be one of the groups that's in the Protist tournament that we get to in class. Okay, so let's start with a little talk about biodiversity in general. So the first concept for biodiversity of a group that we cover is prokaryotes, and protists are the next one. And if you remember from the prokaryote concept, I said that most living things are small. The great majority of life is small, even microscopic, and the great majority of living species are probably going to be very, very small things. So let's keep thinking about small things here in this protist concept. One thing that's important, at least considering most earthly life as we understand it, is liquid water is needed for things to live. And so things need to live in an environment where they have access to water. They might need to be in water. Things that are small, because they have a high surface area to volume ratio, which you hopefully remember from the graphing handout, things that are small have problems with exchange. And one of their problems is that they might have too much water exchange based on their small size, based on the surface area to volume ratio. And so things that are small need to be in some sort of aqueous environment. Bigger things, as I say here, have evolved some sort of mechanism. Plants evolved several ways to get away from water. Even though animals are filled with aqueous extracellular environments, and of course the intracellular environment is aqueous too, a lot of animals have figured out pathways out of the water, but we'll cover those when we cover animals. But in general, things that are small and all cells need to be in some sort of aqueous environment. So let's kind of think for a second. Let's come up with a list of ways of different environments that are aqueous that things can live in. And what I'll invite you to do here is actually pause the recording and make yourself a little list. Okay, so hopefully you all have made a list, and let me kind of make one of my own lists. So what comes to mind first? I'm guessing most of you, the first thing on your list is the ocean, rivers, lakes, other wetlands, streams, creeks, probably every version of that you probably have in your list. Puddles. Puddles are an aqueous environment that's habitable. But one that water droplets. So but one that you probably don't think about that is a very habitable environment is in or on another creature. So let's talk a little bit more about that. So it turns out that organisms, creatures, species, whatever you want to say, are good environments for small creatures to live in. On the inside, they have nice aqueous extracellular spaces. They might have aqueous vessels. The interior of an organ, the lumen of an organ might be mostly aqueous. The inside of cells, of course, is aqueous. So larger living things tend to be, living organisms tend to be a pretty hospitable environment, as long as the immune response and things like that aren't too big of a problem. If there's a way to kind of counteract that. Think about the other things. Think about the protection that small creatures get by living in or on bigger creatures. They're going to be more protected from the environmental fluctuation. They'll be protected from their own predators, their own pathogens, and even small things can have pathogens, as we'll cover. And if you remember that viruses even have their own pathogens, the variophage viruses. Living things tend to have lots of resources. So I say fuels and building blocks, the carbs, lipids, proteins, minerals, vitamins, other chemicals that are necessary for life. So what's the better, if you're a living organism that needs to absorb all those things to maintain yourself and survive, what's a better place to find that than in a cell or a tissue? Or a vessel that an organism uses to transport things. So large species tend to be wonderful places to live. So because of that, biology is filled with symbioses. So we covered symbioses a fair bit in the prokaryote concept when we were talking about the human microbiome, but symbioses are widespread in other things too. So yes, lots of prokaryotes are symbionts. All viruses are symbionts if you consider them living things, but they are so widespread that they show up in every group of organisms: plants, animals, fungi, prokaryotes, protists. They all have lots and lots and lots of symbioses. And it's a pretty straightforward definition of a symbiosis. It's just a shared relationship where organisms live together. They obtain nutrients together. They have a social relationship if they're an organism that actually has measurable behavior. So think about the definition of the term. Sim is same, biosis means life, so same life. We'll talk more about the outcomes of symbioses when we cover fungi, but a symbiotic relationship can be very beneficial to both, it can be negative to both, it can be neutral to one or the other. In general, a relationship, a symbiosis, in general won't be neutral for both species. One species will generally get a positive or negative outcome. And a symbiosis is not often going to be negative. That doesn't help anybody very much. But any combination where the relationship can be positive to one species, negative to another, positive to both, all those possibilities exist. An endosymbiont. An endosymbiont is a symbiont. It's a creature that lives in another creature. So there's all sorts of endosymbionts. And by the way, the ectosymbiont is a creature that lives with the host but not inside of its body. I have the nice photos here showing two different endosymbionts from pop culture. Thank goodness neither of them is real. In Quato's case, we're lucky he's not real because he's disgusting. The picture on the right is a endosymbiont of Quato living inside a human. And of course the alien larval stage from the Alien movies is a endosymbiont too, and it's a pretty nasty endosymbiont because it results in the death of its host. And by the way, Quato sort of results in the death of his host too, not to spoil Total Recall for anybody. But that's a negative relationship. So if you think of the alien, the symbiotic relationship would be positive for the alien larva and negative for the human host or other animal hosts. Remember, the host could be the predator, the host could be all sorts of other things. And by the way, endosymbionts can live inside the tissues of a host, the extracellular spaces, but there's even cellular exosymbionts too, or endosymbionts too. Although cellular endosymbionts tend to be algae, prokaryotes, or viruses. A symbiotic relationship isn't necessarily as simple as two creatures living together. In a lot of cases, symbionts use multiple hosts in order to complete their whole life cycle. So in order to complete the entire life cycle associated with reproduction, they might need two different host species, or at least host individuals, to survive. We call this a complex life cycle. And in the case of a complex life cycle, there might be a free-living stage, there might be a parasitic stage, there might be a stage that's a symbiont, beneficial symbiont, mutualist, and a free-living stage. There might be multiple pathogenic stages. So a complex life cycle only means that different stages of the life cycle live and grow in different places. One thing that's really interesting about complex life cycles, I'll even go as far as to say, for the most part, the sexual and asexual phases of the life cycle are going to be found somewhere different. So there might be an asexual free-living stage and a sexual reproductive, there might be a sexual parasitic stage, for instance. There might be a asexual parasitic stage and a sexual free-living stage. Any combination is possible. We tend a lot of times to have an asexual pathogenic stage in us, and a sexual, not really pathogenic stage in another organism. For instance, the Plasmodium parasite, the causative agent of malaria, actually has an asexual stage in us that's extraordinarily harmful, and a sexual stage in the mosquito vector that's not very harmful to the mosquito. Not only are symbionts extraordinarily common and extraordinarily important today, but we wouldn't even have eukaryotes without symbiosis. So let's kind of talk about where eukaryotes came from. So the first evidence for eukaryotes are thought to be just under 2 billion years old. So that's what the BYA here stands for. The majority of eukaryotes that we have today are a little newer than that, but the great majority of them showed up somewhere between 1.5 billion and 1 billion years ago. So where did eukaryotes come from? So hopefully you remember the definition of eukaryote is an organism with a well-defined nucleus and membrane-bound organelles. So one of the first steps was the formation of the nucleus itself. So in this case, the nucleus, in this picture that you see here, this nucleus, which is figure 25.10 of your book, by the way, this figure kind of shows the hypothesis that the prokaryotic DNA was compartmentalized, and that compartment would become the nucleus. There's also some really interesting ideas that the nucleus came from a symbiont, maybe even a virus, but that's not really covered in this figure, but it's just kind of a fun hypothesis to think about. So one of the really important features of almost all eukaryotes, because there's a couple protist exceptions, but almost all eukaryotes have mitochondria. That's one of the most important organelles that makes a eukaryote a eukaryote. And mitochondria wouldn't exist unless there's a symbiosis. So there was a bacterium of some kind, as you see here is an aerobic bacterium, some sort of proteobacterium, according to the genetic evidence. But that bacterium became an organelle. And one of the important things about that is that organelle was put in a specialized, or that ATP production was put in a specialized part of the cell, and that allowed other parts of the cell to specialize in other functions. And that really did a lot to help out the diversity of cells and cause a lot of adaptive radiation in cells. And by the way, one thing while I'm talking about mitochondria here is be really careful. Don't say that mitochondria make energy. So nothing makes energy. Not the sun, not anything. Not 1 joule of energy has been made since the Big Bang. So be really careful about how you say that. Also, symbiosis is responsible for the presence of chloroplasts. Let's look at that a little more. So eukaryotes wouldn't exist unless bacteria were absorbed by the first basal eukaryotes. So there's very strong genetic evidence that both of the important organelles that are unique to eukaryotes, mitochondria and chloroplasts, there's very clear evidence that they are the descendants of bacterial cells. In the case of chloroplasts, a cyanobacterium was engulfed by a early on heterotrophic eukaryotic cell. And this is a diagram here from How Life Works. And what this diagram shows is a phylogenetic tree of cyanobacteria. It's a nice tree because it shows that they're almost monophyletic. If you don't include chloroplasts, they're monophyletic. If you do, they'd be, that's right, they'd be paraphyletic, which you all kind of silently said to yourself right there. So this tree shows that chloroplasts evolved from a lineage of cyanobacteria that became chloroplasts. So there's only very limited numbers of origins of chloroplasts being absorbed. A really interesting thing to think about then is if eukaryotes are more closely related to archaea based on their genomic DNA and based on a lot of their other cell characters, the chloroplast or mitochondrial organelles are actually more closely related to bacteria. So interesting thing to think about is eukaryotes are kind of a chimera, however you want to say it, of archaea and bacterial characteristics. So that's kind of a fun thing to think about. So who are all the eukaryotes? So this is a tree of all the eukaryotes. So based on kingdoms, sometimes phyla, things like that. A lot of these groups is kind of hazy whether they're a kingdom or phylum, but we can just call them taxa. So this is all the eukaryotes. And the first thing you'll notice is that pretty much everything on this tree is a protist. The only exceptions are the plants, which are kind of hiding about 3/4 of the way down, and then way at the bottom, fungi and animals kind of hiding at the way down here too. So most groups of eukaryotes are considered protists. They're kind of freaky, they're kind of messy. So in general, here's the definition of a protist. A eukaryote that is not a fungus, a eukaryote that is not an animal, a eukaryote that is not a plant. If it is not one of those groups, it is a protist. Okay, what else do we want to look at here with this tree? So you'll notice there's 4 supergroups. In the 9th edition, the Rhizarians comes off as a separate supergroup, by the way, but according to the 10th edition, the SAR clade is a nice monophyletic group. So we have 4 groups. We have the excavates, we have the SAR group, or if you want to call it the chromalvelates and the Rhizarians. If you have the 9th edition, that's what those groups are, and the stromatopyles and the alveolates combined are the chromalvelates if you have the 9th edition. The archaeplastids. The archaeplastids is a pretty easy group. The archaeplastids are plants and things like plants. Plants and algae are the archaeplastids. And the uniconts, that's a group of consumers. It's a group of pathogens. It's a group of predators. It's a group of saprobes. And it is the group that includes animals and fungi. So if we look at these different groups, so the excavates, let's look at them from a standpoint of how they obtain their nutrition. So diplomonas and parabasalids are symbionts / parasites. Diplomonas are somewhat free-living. Euglenozoans are going to be mixotrophs. We'll cover that in a bit, but they're photosynthetic and consumers. The SAR clade has a whole bunch of creatures that are both. So the diatoms, golden algae, brown algae, those are all photosynthetic. The dinoflagellates are photosynthetic. The ciliates are photosynthetic if they use a symbiont. The apicomplexans are not photosynthetic. They're completely parasitic. The forms and radiolarans are both consumers. The cercozoans are pathogens. The archaeplastids are all photosynthetic. So we see some of these groups are a mixture. So the excavates and the SAR clade, a mixture of both photosynthetic, free-living and parasitic, everything you can imagine. The archaeplastids and the uniconts are a little cleaner in that they're all producers. So one thing you'll notice looking at that quick survey is that there's a lot of photosynthetic groups of eukaryotes. A lot of different protists are photosynthetic. And an interesting thing to consider is the reason for that is that the symbiosis that led to photosynthesis happened in a whole bunch of different groups. So several rounds even of symbiosis happened. So eukaryotes did not invent photosynthesis. There are no eukaryotic groups that are photosynthetic on their own. All photosynthesis in eukaryotes depends on the remnants of a bacterial symbiont, and that bacterial symbiont would become a chloroplast at some point. So There was a heterotroph that absorbed cyanobacteria. That cyanobacterium became a plastid, a chloroplast. And the descendants of these first eukaryotes that absorbed cyanobacteria became either red or green algae. And it turns out that a lot of these protist groups are going to be, that are mixotrophic at least, a lot of them are going to be photosynthetic because they absorbed an alga, and we call that secondary endosymbiosis. So that is the absorbing as a symbiont an organism that has already previously absorbed a symbiont. And this happened several times. So the red alga was absorbed by the ancestors of the straminopiles and the dinoflagellates. And dinoflagellates and straminopiles are both members of the SAR clade. In fact, straminopiles is one of the large monophyletic groups within that. The dinoflagellates are in the alveolate group. And the descendants of the green algae were also absorbed multiple times with rounds of endosymbiosis too. So, in particular, as for our purposes, they were absorbed by euglenids, and so they became a plastid in the euglenids. And the euglenids are an excavate, which is very distantly related from the SAR clade, which are the other groups of protists that acquired photosynthesis from an alga. Okay, so we need to survey a lot of protists. So on the next slide, I will have a nice list of a whole bunch of protist groups. And if it's in green, it's the one that I want to cover. So we need to know some important information about them. But a better question, other than what you need to know, is which protist is the best. So one of the things I like to do every semester is hold the protist tournament. So the idea of the protist tournament is have students talk about their protists, and they do have to say some very specific things about each. And then the classmates will vote on which one is the best. And what makes them the best? We'll talk about that in class. But let's talk about the protists that you need to know just in general for Biology 1108, both for your lecture quiz and also for the lab and lab exam. You'll see that we have a pretty fearsome list of protists that we need to cover for the tournament. So the ones that are highlighted in green are the groups that you need to know about in some sort of lecture, in lab, things like that. So these groups might be a phylum, they might be a kingdom. It's a little bit rough with higher level classification, especially at the level of kingdom. So we'll just call these protist taxa. Alright, so let's start with the excavates. I'll give a little discussion of each of these things for you, and you can also cover these things in your tournament. So the excavates is our first supergroup, and it includes three major taxa that are worth knowing about. The diplomonids, parabasalids, and the euglenozoans. And the euglenozoids actually contains two smaller taxa that are important to know. Alright, so as far as each of these is concerned, so the diplomonids are going to be, let me back up. So the excavates are all unicellular too. So the diplomonids might be free-living, they might be parasitic, they might have part of their life cycle in either form. They're all consumers. Parabasalids. Parabasalids are pretty easy. They're all symbionts. So they might be gut symbionts of arthropods that helps them digest digest more complex molecules like carbohydrates. They might be pathogens. For instance, there's some pretty nasty parabasalid pathogens of people. The euglenids. The euglenids are all free-living, and they're mixotrophic. So they're going to be small, flagellated, mixotrophic creatures. So they'll have a chloroplast in them. Kinetoplasts. Kinetoplastids are all pathogens. So they're all pretty nasty pathogens too. What makes them so nasty is that in general there's no cure once you have a kinetoplast genetic disease. So definitely want to stay away from those. So trypanosomes, for instance, are pretty well-known kinetoplasts. All right, so let's move on to the SAR clade. This includes 3 major taxa, the Strammonophiles, Alveolates, and Rhizarians, and each of those contains a couple other important taxa for you to know. So let's start with the diatoms. Diatoms are going to be unicellular, aquatic, either freshwater or marine. Diatoms are very cool because they're a glass shell, which in the case of a protist we call a test. We prefer the term test to skeleton. They're all photosynthetic. The brown algae. The brown algae are important because they are protists that are multicellular. They're also, by the way, unicellular. They're also photosynthetic, and they are going to be predominantly marine. In the 10th edition, there's a very sad missing creature in the oomycetes, or in the Strammonophiles, and that's the oomycetes. So the oomycetes are aquatic, fungus-like, multicellular protists. So these are predominantly pathogens or saprobes, so they're decomposers, very much like a fungus, and they're aquatic unless they are a pathogen of a terrestrial organism. So sadly, sad to see them go, but I like them too much to take them out of the tournament, so we're still going to cover them. Note that the Strammonophiles have lots of multicellular protists, so protists are not necessarily unicellular. Okay, moving on next to the alveolates. There's 3 groups in there that you should know. The dinoflagellates are unicellular mixotrophs. So most of them are photosynthetic, but they are also going to be often predatory, or at the very least they can use phagocytosis to absorb other microbes. So they tend to very often be found in marine habitats. Sometimes they might be found in freshwater habitats too. Apicomplexans. Apicomplexans are all pathogens, all unicellular pathogens. Generally have a complex life cycle. Malaria is caused by an apicomplexan. Toxoplasmosis is caused by an apicomplexan as well. So very amazing little pathogenic creatures. Okay, ciliates are the largest of the unicellular protists, named after, of course, the cilia that they use for either locomotion or feeding. And they are mixotrophs, so they tend to have algal symbionts, and the great majority of them can also use phagocytosis to consume other microbes, and they live in aquatic habitats. Although there are some gut symbionts that are ciliates also. Okay, the rhizarians, those are going to be 2 single-celled, or at least the 2 that we cover. There's actually a third one. But the 2 that we cover are going to be single-celled marine predatory creatures for the most part, although they can have algal symbionts. The rhizarians, the forams, and the radiolarians are both pretty cool. They both have a test. So they both have a little shell that's very useful in aging, in dating, in looking at climate change, in oil exploration. And in the forams, the test is made out of calcium carbonate, and in radiolarians, the test is made out of glass. It's silica-based. Okay, archaeplastids, that's another nice monophyletic supergroup, and that is red algae, the 2 groups of green algae, and plants. So we'll cover the archaeplastids quite a bit more in the plant concept. And the last group. The last group is the uniconts. So the uniconts are a very important group of eukaryotes, which includes several prokaryotes, and it includes the animals, it includes the fungi. The only uniconts that we're really going to cover are going to be the amoebozoans in terms of protists. So amoebozoans can include amoebas, of course, and it can also include slime molds. And so slime molds are going to be multicellular amoebozoans. At least the amoebas aggregate into a multicellular structure with either kind. And they're going to be mostly saprobes. In some ways, they can feed on microorganisms too. And the amoebas, they're going to be more single-celled amoebozoan, little blob creatures. They can be found all over the place. So slime molds are generally terrestrial, but amoebas can be aquatic, they can be terrestrial, they can be symbionts. They can live inside all sorts of animals, including us. Amoebas can be pathogens, saprobes, kind of predatory, as protists can get. So there you go. In just a little under 10 minutes, that's the diversity of protists, but you'll definitely want to read quite a bit more. You'll definitely want to study them up for your presentation, and we'll be covering each of these groups in class and in lab as well. And you'll get to see them in lab, or at least see many of them in lab, and we'll discuss the great majority of them in class as well. All right, so I said the majority of these things already. So these are the things that you should know. I don't want you to know excruciating details. I don't really care about the anatomy or what the word uniconta means. But for all the groups that are in green, you should know kind of these basics. So you should know where they're found. So are they a marine organism? Are they a single-celled marine organism? Are they planktonic? Do they float? Are they attached? Are they sessile? Maybe they're a symbiont. How does the organism get its energy? So does it produce its own fuel? Is it a producer? Is it an autotroph? If it's a heterotroph, if it's a consumer, is it a predator, which I'll say in quotes? Is it a saprobe, which is another word for decomposer? Is it a parasite or pathogen? So you should know those things for each group. And of course, those things can be very different too, or be very variable within a group. So one group, for instance, the amoebas, has everything except producer. All the mixotrophs, of course, are producers and consumers. So do be aware of those. And then we're going to want you to know something, if they're important to us in either a good way or a bad way, how are they important to us? So what are some uses of this protist? What are some benefits we get from this protist? Is this protist harmful to us? As you can guess, most of the pathogenic protists are going to be harmful to us. Okay, let's talk about the ecology of protists for a little while here. So producers can be pretty much fitting any of these ecological roles, especially if you're looking at each of these 4 roles from a trophic point of view. I am going to take a second and define the term predator and saprobe here, since these are kind of coming up for the first time. So it's kind of wonky to call a protist a predator. I know it's kind of a little weird. But when you read about protists, you might see the term predatory. So that is an act of feeding. One act of feeding will consume one prey organism. So when we think of predators, we generally think of a wolf eating a deer. We generally think of a cat eating a bird, or a bird eating an insect. But it's not necessarily, that's the carnivorous viewpoint of a predator, but we can also have herbivorous predators, believe it or not. So a predator could potentially be eating seeds or eating flowers. So herbivory that kills and eats only, or kills and consumes only flowers and seeds, that's predation. In the case of small creatures, so planktonic, filter-feeding creatures, So there's plenty of protists that are predatory to microbes. So we'll cover those a little more. What's a saprobe? A saprobe is another way of saying decomposer, carrion feeder, things like that. So something that removes waste. So we'll spend a moment trying to figure out how each of these affects humans. So let's talk about each of these four roles. Okay, so what's the probably primary importance of photosynthetic protists? So first of all, they're very important parts of food webs. So phytoplankton is a term that includes protists, algae, cyanobacteria are part of the phytoplankton. So these small floating producers are important bases of food webs. And so producers are part of that. In particular, protists are part of that. In particular, things like diatoms, dinoflagellates. Those are huge bases of food webs. Another important thing for us in terms of photosynthetic protists is their ability to produce oxygen and, of course, their ability to remove CO2 as well. So let's talk about the importance of that. The most important thing is our diatoms. So diatoms are a huge part of the phytoplankton communities in the oceans and in lakes. So diatoms are, when it comes to preventing global warming by removing CO2 from the atmosphere, it turns out they are as useful as a tropical rainforest. So while we really think of, oh, we gotta save the trees, save the rainforest, we also should be thinking of saving these beautiful little creatures, these diatoms. And by the way, the top image is of a diatom, very beautiful. The bottom image is of a dinoflagellate. And of course, diatoms, dinoflagellates, not only do they remove CO2, but they also produce oxygen, and so they are important parts of our oxygen supply. And protists are very important too if they put carbon dioxide in their test. So let's talk, or if they take carbon from the atmosphere and put it in their test. So let's talk about that in a little bit. Okay, there are producers. There are protists that we eat. So I was saying these are a very direct relationship. So there are protists that are photosynthetic that we eat. What's the most obvious one? Kelp. So kelp is a multicellular, very plant-like protist. Interestingly, it's not very closely related to plants. It's not even monarchiplastid, but it looks a lot like a plant. It acts like a plant. It has a leaf, quote-unquote, stem, quote-unquote, roots, quote-unquote, but it doesn't have those actual parts. It has structures similar to those. So we eat them. I've eaten kelp. Most of you have probably eaten kelp. These protists are very important symbionts. So algae tend to be very important symbionts of other things. So lots of protists have algal symbionts. The rhizarians do, ciliates do. Lots of animals rely on algae as symbionts as well. The one that I'll discuss that's most important is a type of symbiont called zooxanthellae, or members of the dinoflagellate genus Symbiodinium. So Symbiodinium is a endosymbiont of corals, and corals, as we'll cover in a couple concepts later on, are animals. And they have a very important symbiotic relationship. So the corals, of course, protect the algae, and the algae use photosynthesis to provide food for the symbionts. So this is kind of one of the really standard symbioses that we see in nature, that we have a photosynthetic symbiont getting protection from a consumer host, or at least a consumer symbiont. The zooxanthellae are very important. So if the corals are stressed, particularly by things like temperature, water warming, they will boot the zooxanthellae out of them, out of their little colonies. And when that happens, we call that bleaching, because the color from the photosynthetic pigments in the algae is no longer there, so the coral appears white. And if they don't regain them, they can starve. So think about what it means to have corals. So without zooxanthellae, just with these communities getting warmer, when the corals lose the zooxanthellae, they can die off. They can be dying from starvation. They can die because they are more susceptible to diseases. And there's actually a marine biologist at UGA that does phenomenal research looking at coral diseases. So it's very bad to be bleached. And it turns out that the coral reefs are a very important part of marine biodiversity. So in the oceans, at least, they are the most diverse ecosystems on Earth. And as far as sheer animal phyla are concerned, and sheer diverse groups of animals, they're actually the most diverse ecosystem on the planet. They are sources of amazing drugs. They are sources of food, because we harvest some of the animals that live in the coral reef. With drugs, we're getting lots of cancer drugs from corals. So that's actually why I have this great book here on the PowerPoint, Sex, Drugs, and Sea Slime, which talks about all the drug discovery that can come from coral reefs. We definitely want to lose them. So there's so many ways that we benefit from corals, and thus we need their healthy symbionts. It turns out that even the remnants, the remains of phytoplankton, of photosynthetic protists, is very important to people. So first of all, a lot of fossil fuels are derived from phytoplankton. So for instance, most of the world's oil actually comes from plankton that were around a very long time ago. So even though we might think oil comes from dinosaurs or something like that, it actually doesn't. Diatoms are tremendously useful for looking for being silica. So diatomaceous earth can be used for all sorts of things. So it's used as a chromatography filter. Those of you guys that are using taking chemistry might well use diatomaceous earth, or at least some sort of silica bead for it. Diatomaceous earth can be a type of insulation. Diatomaceous earth is actually, very sadly for me, an insecticide, although it's a non-toxic insecticide to people. The way diatomaceous earth works for insecticides is that the tiny little silica grains actually will scratch off the protective outer layers of the insect cuticle, and they die pretty quickly from desiccation, because they have an open circulatory system. So diatoms are tremendously useful as far as diatomaceous earth. And it turns out that even the skeletons, or the tests, which is a better way to put it, of other protists are going to be pretty useful too. So for instance, the rhizarians, even though they're not producers, I'll throw them on here in this slide where we talk about shells being useful. So the tests of rhizarians, in particularly forams and radiolarians, they are great for looking at prehistoric climate. They're great for understanding climate change, things like that. Let's move on to protists that are consumers. So some protists are called mixotrophs. A mixotroph is just a mixture of autotroph and heterotroph. So it's an organism that has the ability to make its fuels and also can obtain fuels and building blocks from the environment. So there are some protists that are mixotrophs that use plastids. So they have their own chloroplasts that are derived from engulfing a cyanobacterium. So the euglenids, for instance, have chloroplasts. The dinoflagellates have another type of plastid, but they still can do photosynthesis on their own. And dinoflagellates are considered themselves algae because of that. But there are also some groups of protists that are hosts of algal symbionts. So the ciliates are an example of this. The ciliates can be mixotrophs because they absorb algal symbionts and use the products of their photosynthesis for their own nourishment. By rhizarians, I am talking again about the amazing forams and radiolarians. So both of them may use algal symbionts as well. So the first completely heterotrophic type of protists that I'll discuss here are the pathogens. So pathogens, agents of infectious disease, the causative organisms of infectious disease. So there are lots of protists that are pathogens that are bad for us. So not just wildlife pathogens that bother us, but they can be pathogens of humans, they can be pathogens of our pets, they can be pathogens of our crops. So one of the most well-known, and I would say most people would agree, the deadliest of all infectious diseases and most prevalent infectious disease is malaria, caused by 4 species of Plasmodium. There's actually quite a few more species of Plasmodium. There's just 4 species that attack humans. I actually have this photo on the bottom left to kind of remind myself of malaria. This is for a movie recommendation. So there's a documentary, at the time of recording, this is streaming on Netflix, a documentary called Guns, Germs, and Steel, which is based on Jared Diamond's book of the same name. And one of the segments of the documentary shows him visiting malaria patients in Africa. And this screenshot I took here, Dr. Diamond is actually breaking down seeing all of the malaria patients, particularly the kids. And so it's a very powerful, very moving, and a great documentary. So I just have this on here to recommend it. The oomycetes, the poor oomycetes left out of the book. So they're very important pathogens. So there's a blight caused by a oomycete called Phytophthora infestans. And Phytophthora infestans attacks plants in the potato family, including, of course, potato itself. And imagine the kind of social, political ramifications of that little oomycete causing the starvation. So pretty important little creatures there. This is a good time to bring up some very important routes of infection for pathogens. So 2 of the most common routes of infection are oral route, which is consuming an infective stage of the pathogen. That could be either from food or water. And the vector route of infection. So a vector is a living thing that is going to transmit a pathogen, an infectious disease, from host to host. Let's take a look at these 3 pictures here. So on the left we have a tsetse fly, which is the vector of the trypanosome that causes African sleeping sickness. In the middle picture we have the kissing bug, which is the vector of the trypanosome that causes Chagas disease. And on the right we have the sandfly, and that vector is the trypanosome that causes leishmaniasis. So all 3 of these are biological vectors, and they are vectors in which the sexual phase of the pathogen's complex life cycle happens in the vector, and the asexual disease stage is in us. So all 3 of these nasty little insects can cause some pretty nasty diseases None of these have a cure, by the way. Sleeping sickness, Chagas disease, leishmaniasis, there's no cure for any of them. Plasmodium is also vectored by mosquitoes, the causative agent of malaria. Okay, I want to talk a little bit more about predators and kind of be ready for this. You'll see this on the lecture quiz. So the idea of protists being predators. So it's kind of a weird, wonky definition of predation. So and what I mean by that is one microbe using phagocytosis to consume another microbe, is that considered predation? Technically, yes. So an act of phagocytosis is going to kill one microbe, and that microbe will be consumed. So technically, things like amoebozoans are predatory. Dinoflagellates are actually often predatory. Using this definition, the radiolarians and the forams are all predatory. So do think that through. It's kind of interesting. As far as I've been talking with a lot of these other protists about what they mean for people, these don't do too much. The protists as predators, dinoflagellates can actually kill off fish and make them toxic to people, but in general, the act of predation doesn't really, by protists at least, doesn't really do too much. Later on in the class, we're going to talk about animal predators. That can be tremendously important when we talk about biological control of pests. Okay, the last ecological role of protists, the saprobes. So remember saprobe. It's the nature's garbage disposal. It's a organism that is decomposing. So protists, bacteria, fungi, lots of animals, they have an important job. Their job is to get rid of garbage. Whether they're talking about dead bodies, whether they're talking about feces, what have you. Lots of organisms are used to clean that up. So the slime molds, like you see the picture on the top, the slime mold is an important terrestrial saprobe. The saprolagnia, the oomycete, is actually an important aquatic saprobe. Two of the most important types of saprobes are, as far as protists are concerned, the oomycetes and amoebozoans. And it turns out they're very similar to fungi. So a slime mold looks like mold. A water mold looks like mold. Heck, just look at the common names of the oomycetes and amoebozoans. Slime mold, water mold. They look like mold. They look like fungi. And it turns out protists are very, very important saprobes in the water. So the water doesn't tend to have as many fungi as terrestrial habitats. So who takes over? Who does the job of fungi? Yes, water molds and amoebas do. Okay, so that's about the end of protists. I'm going to leave you with a question here. Alright, so here's a kind of a symbiont / interaction ecological question that I want to pose to you guys and give you some time to think about. Is a saprobe a symbiont? So what's the definition of a saprobe? It's a decomposer. In general, by the way, we're talking about a microbial decomposer. What's a symbiont? It's an organism that lives in association with another organism. So a question for you all to think of, are saprobes symbionts? We'll cover this more when we get to the fungi. Alright, so this concludes the protist lecture. Hope you all enjoyed it, and I wish you all good luck on the lecture quiz.